Well, welcome for those who have been able to join us today for our monthly AMS seminar. My name is Jonathan Plett. I am the president of the Society. And thank you very much for being here. And I'm very pleased to introduce our speaker this month, Dr. César Marín, who is from the Santo Tomás University in Chile, where he works in the Research and Innovation Center for Climate Change. Now, César has a hugely impressive list of things he's involved in and research that he's done. He's the leader and founder of the South American Mycorrhizal Research Network. He's worked tirelessly in shining the light on aspects that we don't understand about mycorrhizal functioning and mycorrhizal diversity in South America. And that's been really pleasing to see how he has been really a leader in that area of uncovering some really cool stuff. He's addressing these gaps, especially if you look through the different publications that he's been doing in the last years, looking at functioning of mycorrhizas as well as their diversity, both in natural settings, but also in more agricultural, like chocolate farming, which I find really cool. So today, César is going to be giving an overview of his work in Chile, but is also going to be talking about the South American Mycorrhizal Research Network and how we can all get involved. With that, I will pass it over to you, César. Thank you.
So hello, everyone, and thank you, Jonathan. And also thanks to Camille Tron for the nice invitation to this seminar. So the introduction by Jonathan was quite good. I don't have anything to add. So the title of this presentation is Southern and Global Mycorrhizal Collectives, and you will see why. First, I will talk about my research over the last more or less 9 or 10 years here in southern Chile. It's northern Patagonia. Then on this concept of mycorrhizal collectives, which I will explain in detail. And then about the South American Mycorrhizal Research Network. So first, Southern Chile mycorrhizas. Well, this slide is always for the Northern Hemisphere people because they are always surprised that we have a native arbuscular mycorrhizal fungal associated conifers. So, or 10 conifers or 10 native conifers that we have both in Chile and in Argentina are AMF, while the dominant tree, which is the Nothofagus species, or the Nothofagus genera, sorry, comprised also by 10 species, is ectomycorrhizae, or ectomycorrhizal. So in this chapter, we analyze the roots from 245 species, very common in several forest types, and more than 80 or almost 80% of the vegetation is associated to Glomeromycota, because in many of our countries we don't have this really basic information about mycorrhizal types. So I think that's a very important first step. I have been studying biodiversity with several methods, including just like classical morphological identification. In this very short study, in this short article, in Puyehue National Park, we visit the park for 5 years each season, and found a total of 26 fungal ectomycorrhizal species. Most of them Cortinarius. Cortinarius is a really common genera. Here in Chile, we have around 300 or 350 species of Cortinarius. And well, we also did the root staining to see which mycorrhizal types are present. And not just ectomycorrhizae, but also we have been studying the morphological biodiversity of arbuscular mycorrhizal fungi, because until this paper from 2017, most of the studies, or all the studies in Chile, were mostly focused on agricultural settings, but very few studies were conducted in forests. So we went around like some 20 forests in in different regions of Chile and study the biodiversity based on spore analysis. So this study in particular implied analysis of, I think, 90,000 spores, so a lot. Sadly, we don't have too many taxonomies. Here in the slide is Paula Aguilera, who is, I think, the only taxonomist in this group, in this very important group. And something very interesting is that about 1/3 of the species are shared between the native forest and those agricultural settings. Then also in 2017, we published this article with pyrosequencing, which was one of the first studies here in Chile using this technique. And we did a basic analysis of land use change effects on different fungal guilds. So basically finding that in pristine forests from Nothofagus pumilio, these forests are pristine, are very, very nice forests, like 500-year trees, and no management of the trees. We found higher abundance and richness of both saprophytic fungi and ectomycorrhizal fungi. When we increase it, or when the land use was more severe, when there were more disturbances, these both guilds decreased a little bit, and plant pathogens increase both their abundance and their richness. So this was one of the first studies. Then this year, recently, I published this other study with Illumina MySeq sequencing, and in that study we compare the two mountain systems that we have. It's not only the Andes, but also the coast mountains, which are very important, and which were like a biodiversity refugia during the last glacial maximum. And because of that, the plant biodiversity is way higher in the coast mountains than in the Andes. So we compare these two mountain systems and the Nothofagus forest and the native conifer forest in 5 national parks. Basically, the result of all of this is that saprotrophs were way more abundant and way more diverse in the native conifer forest compared to the Nothofagus forest. So we suggest that maybe the litter of these native conifers is quite complex, is difficult to degrade. Also, for example, for Araucaria, Araucana, the leaf can be 27 years in the tree, and after 27 years, it falls. So maybe it's related to that in the case of saprotrophs. And well, as expected, of course, ectomycorrhizal fungi were more abundant in the Nothofagus forest. I will talk a little bit, just a few slides about the main topic of my PhD thesis, which I finished almost 5 years ago. The topic was biogenic weathering, which is this process by which, by direct or indirect action of the IFE, in this specific case, the mineral layers are degraded, are subject to this weathering process. So this is a big research agenda, almost 25 years. Different teams around the world have been studying this process. There are now very, very good examples in situ, because when I started this, most of the examples were in laboratory, but now there are very good examples in situ. And what we did was to bury these mineral pellets of muscovite and biotite, which have the cations in red there, so potassium and magnesium. And we bury we put these bags 15 centimeters soil depth in the same plots of the Illumina MyCiiC study in the same exact plots. And then, well, of course, the pellets are designed in order that ivy enters and something smaller than ivy, but not roots. And well, it was a lot of work, of course. It was putting the bags in these 13 forests, collect them 1 year later and 2 years later, and then analyze this in the microscope, mainly XPX microscope, and also confocal. So the photos reflect, the photos are a proxy, the colonization on each photo, the colonization of the ivy constitutes a proxy of this process of biogenic weathering, because it's mineral that now no longer belongs to, is a part of the mineral particle which now is in the soil. It is not longer in the mineral. So we calculated the colonization of each photo as a proxy of biogenic weathering. So one of the main results, or the main result, I would say, is that there was a negative or an inverse relationship between the colonization of these minerals, biotite and muscovite, and the availability of potassium in this case. We combined the data from Chile and New Caledonia, because this experiment was also conducted in New Caledonia. They have very similar flora to us. And there was a really nice negative relationship. Other project that I am still analyzing the data is these gradients that we find particularly in Nahuelbuta National Park, in these coastal mountains that I mentioned before. It's a wonderful park. It's like an island, basically, because it's surrounded by exotic conifers. So you walk in the park, it's the same altitude, the soil is the same. I mean, chemically it's almost the same, but in a few hundred meters, in 400 meters, it changes, the forest, it changes from Nothofagus dominated to Araucaria dominated, Araucaria araucana, which is a conifer. So it's very interesting, these sudden changes in few meters, and we are still analyzing some of the results, but one preliminary analysis is that the higher the tree biomass, because it's relatively easy to estimate the tree biomass for Nothofagus and for Araucaria. The higher the tree biomass of Nothofagus, for example, the lower the abundance of ectomycorrhizal reeds. And the same for Araucaria. So the higher the biomass of Araucaria, which is associated to arbuscular mycorrhizae, the lower the abundance, the reeds of arbuscular mycorrhizal fungi. So a very interesting result, is still trying to process what this means, work in progress. In the same park and using the same system, we did a sort of a classical experiment on both in greenhouse and in situ with Araucaria araucana seedlines. So in the greenhouse, these were the main results. We had 4 treatments. One, of course, one non-mycorrhizal control. Then in blue, a single species inoculation with 1 species of Glomeromycota commercial. Then 4 species, a consortia, also commercial. And then 17 species that were extracted from the same source, from the same site as the seeds of Araucaria araucana. And well, in A, B, C, and D, you have like shoot, fresh, white, different weight-related parameters, shoot and root, dry and fresh. So in all cases, of course, the treatment with native fungi had better results. Then in a second phase, this previous one was 1 year and a half, more or less, and then another year and a half. We replanted back these seedlines in the forest. We replanted them in the Araucaria forest and then in the Nothofagus forest, 400 meters apart. And what you see there is that they grow way better in the forest where the seeds were extracted in the Araucaria forest than in the Nothofagus forest. So also this is under revision, and well, yeah, very, very interesting results that speak us about coadaptation maybe, or maybe adaptation to the local conditions, basically. We have done several other experiments with double inoculation, for example, of a 2-vector mycorrhizal fungal, pretty well known, and different species of Nothofagus. And then also a bit of my research recently is more applied, is in the, because here is very difficult to grow things. The soils are very acid, very high aluminum saturation, like 70%, and very low phosphorus, maybe 5, 6, maximum 10 parts per million. So of course we want to use mycorrhizals for these conditions. And along with Paula Aguilera, she created this company, Mycorrhizal, which is the first and only Chilean company using AMF as fertilizers, as inoculants. There are other companies from other countries like Poland or Czechia or Mexico, but this Mycorrhizal is the only Chilean one. And I would say that the secret is to apply consortia, and also one good secret from Paula is that there is a follow-up in the places that she has applied these products. So she continuously analyzes mycorrhizal colonization and extra radical mycelium and several other parameters in these places that buy the product. So that's a good secret of the company, of the success of the company. Currently, we are implementing a project to expand, because this work is mostly in southern Chile, but we want to expand to the central part of Chile to wine production, because of course wine is a huge product here, and also because the vineyards are more at the south of the country. So we want to apply like different inoculants from the central part and the southern part of Chile. And one last thing that I am doing recently, very recently, since last year, is this SPAN project, the Society for the Protection of Underground Networks, which of course it has a lot of media attention, and specifically we sample, which is considered possibly the oldest tree in the world. That in the photo, in the photo, the species is Fitzroya cupressoides, the Latin name, and there in the photo is Jonathan Barichivis, the person who dated the tree. The article of the age of the tree is under revision, so we still cannot say that it's the oldest tree in the world. It's under review, but the proposed age of this tree is 5,458 years. So it's really, really old. In Alerce Costero National Park here, very, very close to where I live. So we sample the tree, and we sample other trees from the same species in the center of the tree, and also in a 5-meter perimeter, and also preliminary results. But basically, we found that the tree is called abuelo, meaning grandfather, because actually the grandfather of Jonathan, which was a park ranger, was the one that discovered the tree like 50 years ago. So we found that the overall soil fungal biodiversity is higher in the abuelo itself compared to its perimeter. We for the abuelo, because it's so huge, we had 5, 10, and 15 meters perimeters. And then also richness, fungal richness is higher than other younger trees from the same species. We here also found a negative relationship between AMF richness and the biomass of the trees, excluding the abuelo. And well, of course, this tree is huge, almost 66 tons and 150 meters of volume. Basically, in Chile, we have been studying soil biodiversity in general, not just fungi. In this work of my student, of Javiera, we analyze general biodiversity of several taxa, bacteria, fungi, archaea, and also several soil ecosystem functions like nutrient cycling and soil respiration, decomposition. So we know more or less what is going on. We know more or less which regions of the country and which taxa and which functions need way more studies. And to end this part, there is also a big effort in public education in this Spanish article, which is directed to park rangers. So the park rangers have like this journal in Spanish. So we talk about fungi in general from when they were studied first, which was Darwin. Then the lack of information in the Ministry of Environment. Then which species have some degree of conservation threat. Also, we mentioned that the fungi is not incorporated in the curriculum, in the public scholar curriculum. But thanks to people like Juliana Furci, co-author in this article, and Fungi Foundation and several other NGOs that work a lot, and several citizen scientists, there is like a lot of love for fungi in Chile. It's a very mycological country, and by law is required the protection of fungi. So now, mycorrhizal collectives. This part of the presentation is related to a question that I have been asking myself for many years, which is what is the unit of selection, of natural selection in arbuscular mycorrhizal symbiosis? Why? Because of course they are so strange. So is it nucleus? Is it hyphae? Is it sporocarps? Is it fungi and the host, or other things? So there are several proposals in the literature. Is a topic that has been researched for several years. For example, some propose that nuclei are the unit of selection, but also nuclei and hyphae. There is a nice recent preprint by Jim Beaver and co-authors showing more or less the same. This from the Toby Kear's group is like a modeling approach, but this preprint by Jim Beaver and co-authors is like empirical evidence that sometimes natural selection acts in the nuclei, sometimes in the hyphae. There was a very nice experiment almost 20 years ago from the Bonfante group in Italy, where they artificially selected several generations of spores, and they show vertical transmission of endobacteria, which is super interesting and is kind of a proof that at least in artificial selection is possible, that natural selection seems to be operating maybe at the spore plus this endobacteria. And of course, they do also these very strange things like these huge sporocarps. Some species like Rhizoglomus maya form these huge sporocarps, which also makes me question what is going on there. So one of the frameworks that can be used to study this is multi-level selection, which has many definitions, but one that I like the most is that basically selfish individuals beat cooperative individuals and cooperative groups beat selfish groups. So this is like the standard definition from Darwin, from the Origin of Men, 1871 book. I have published a bit on the subject and also both like reviews, but also an empirical study in mice, in corn, where I found evidence of multi-level selection in corn. And I started to notice recently that the subject has gained a lot of attention recently. For example, some use a multi-level selection framework for the origin of the central dogma in biology. Others use it for cultural evolution. And one study, very recent study, like from May, that I really like it was this entitled "De Novo Evolution of Macroscope Multicellularity." I think that in Nature, this one, where basically they select, is a classical artificial selection experiment, which there are hundreds and hundreds of these experiments. They selected, they started with lonely single cell yeast, and they selected the bigger ones, and they started to aggregate for 1 year, which in yeast is 3,000 generations in anaerobic conditions. So to some this result, they found evidence of macroscopic multicellularity, and even in the tube, in the lab tube, you can see how this evolved descendants become basically a macroscope or a multicellular organism. So because the subject is again gaining traction and gaining attention, I conducted, or I am doing a literature review on multi-level selection and group selection. Group selection was the older term, but from the 80s, more or less, or 90s, is called multi-level selection. So this is work in progress, but basically, after several filters of the literature, I have around 950 articles about multi-level selection, and from those 950 articles, about 100%, in my view, direct empirical evidence of multi-level selection. One of the ways to measure this in nature, not in experiments, but just in natural populations, is through contextual analysis, which is a form to measure natural selection, very similar to the classical quantitative genetics multiple regression, but adding the effect of the neighbors, of the surrounding individuals, which sometimes are related, genetically related, but most of the times they are not genetically related. And in all these students from 95 to 2023, in several types of organisms, what the authors have found using that equation is that sometimes group selection is bigger, and sometimes individual selection is bigger, has a bigger effect on an individual fitness. So why I mentioned all of this and why this topic? Well, recently there is a lot of research in arbuscular mycorrhizal fungal hypospheres, specifically the role of this bacteria in phosphorus and nitrogen mineralization, but it seems like it's not just any bacteria. In this article, new phytologist article by Wang and co-authors, they found the same stable bacterial gills, but not the same taxa. So there is they make a very nice distinction between the gills and the taxa across vastly different maize or corn crops in China. And the most interesting part is that they also found the same gills and the same units, the same gills, also in microcosm, microcosm experiments. And these bacteria contribute to phosphatase activity. Then the group from Jan Jansa in Czechia found, I would say, similar things, but related to nitrogen mineralization. And they don't mention this concept, but reading a lot of philosophy of biology recently, I found this paper which has a new definition of the hologenome or the holobiome in general. Because the classical definition of the holobiome or the hologenome is is adding the host genome and all the genomes of the microbiome of the interacting microbiome. That's like the classical definition of the hologenome. What Javier Suarez says is that that's a very like relaxed definition of the hologenome. So he proposed that is the host genome and some traits of the microbiome. And in these concepts, it matters more the traits than the taxa. So the taxa can change, but the traits are stable through generations and also through through space. So this is very similar to these studies, at least to the study of Wang and co-authors, which found like this core microbiome. So it's very interesting that people from philosophy of biology is coming up with concepts very similar to what people in mycorrhizal ecology is finding. So in this commentary, New Phytologies, along with Nancy Collins Johnson, we propose or ask if the mycorrhizal collective is a unit of natural selection. We define in that article, in that commentary, expanding on the ideas of Daniel Molter, we define the mycorrhizal collective as the host and traits of some few arbuscular mycorrhizal fungi plus traits of some few hyphosphere bacteria. And of course, this depends on soil nutrients, but that's like the general idea of what we suggest a mycorrhizal collective could be. And then in the same article, we put out there the idea of what we call the geographic mosaic theory, which is ask the question if do plants actively cultivate networks of hyphae to provide infrastructure, or do this hyphae provide habitat for this specific bacteria? So in general, the question is what is the role of a spatial structure created by residual hyphae? Because in Glomeromycota there is proof that hyphae sometimes is there in the soil, not connected to any plant. So it's really curious to see what this hyphae is doing. And then the last part on the South American Mycorrhizal Research Network, and I will try to be as fast as possible. Well, the South American Mycorrhizal Research Network, we are almost 340 members from 46 countries. We started as a network in 2017. why specifically South American, which is a question that people always ask me, because the plants, the soils, and fungi of entire countries and biomes are not well known. We don't know the distribution of mycorrhizal types. We don't have like continental or multilateral funding, so all the projects in our region are national, and we know that ecology is no longer, we shouldn't do ecology just at the national level, but regional level. We also need more training in molecular methods, in bioinformatics, in statistics. And we don't have a full integration, a causal integration between biodiversity and functioning in mycorrhizal ecology in general. So many people study just biodiversity and many people just functions, but there is not like this causal integration. In the figure that I show an analysis that I did on the fungal traits database, which shows that South America has very few AM and ectomycorrhizal fungal species hypothesis, but this is of course not true. It's like a bias on the database on how little have we sequence or fungi here. Then with Guillermo Bueno, I did this other analysis on literature research on literature on mycorrhizals, and found that Europe, despite being almost three times smaller than South America, has almost 3 times more publications in mycorrhizae, and they focus mostly on anthropogenic effects on the symbiosis, while our research focus more on community structure, basically, so biodiversity. Then a more detailed literature analysis, again by my student Javiera Rubio. We found 532 articles and 1,500 sampling sites, but for different types of mycorrhizae: arbuscular, ecto, orchid, and ericoids, and functions. So you here in the maps, you see that there are huge parts of the continent with no sampling points at all. The Amazon, the Pampas in Argentina, most of Paraguay, or almost all Paraguay, northern Chile as well. And there are functions like chemical defense, like allelopathy, like water content, with very, very few studies, as well as ericoids and orchid mycorrhizae. So as a network, we have conducted, or we have done 2 international symposiums. The first one was here in Valdivia when I was a PhD student in 2017. The second one was in Bariloche in Argentina in 2019. A really, really nice symposium by the people in Bariloche, like Nahuel Policelli and Natalia Fernandez and others. Very well organized. And of course, we are doing our 3rd international symposium on the mycorrhizal symbiosis in South America, in Leticia, which is a beautiful city in Colombia, in the frontier with Brazil and Peru. It's possible to attend online. If you want to attend online, it's possible to attend online. And this will be at the end of August, and we will have a couple of courses before the symposium. Well, as a network, we are integrated with many other networks. We collaborate in different things with the Global Soil Biodiversity Initiative, with the International Mycorrhizal Society, of course, with Soilbond, and through the leadership of Marcela Pagano and Monica Lugo, we have published 2 books, 2 Springer books, very nice books on the mycorrhizal fungi in South America. We also have a YouTube channel, almost 40 interviews so far, to many different people on all the stages of their careers. Here, the help from Camille Trong and Guillermo Bueno is huge for this channel. And well, I'm the editor of the International Mycorrhizal Society newsletter, so you are welcome to contribute with these interviews, with short articles. Here is what a newsletter looks like. The 11 issue should be out soon, next month. So we have the top 10 papers of the last 4 months. We have highlights, we have commentaries, interviews, tools, events, etc. And that's it. Thank you.
Thank you very much, César. That was an awesome overview and a lot of great information. For those of you who are attending online, if you want to pop in questions that you may have into the Q&A section of the bar at the bottom, that would be great. And we can read them out to César. While we're waiting for any questions from the audience, I thought I would start. So I found it interesting where you were looking at the arbuscular mycorrhizal fungi in between the native forest and then the agriculture that's happening right beside. Within that, so did you get any impression or any idea of, you know, so you were saying that there are Chilean specific AMF. There's also introduced, from what you were finding, were you finding that there was, you know, introduced AMF that were going into the forest, or did they kind of stay with agricultural, or was it all native AMF that were coming into agriculture?
Oh, that's a good question. Yes, there are some species, but very few species of the understory of introduced plantation of exotic plantations. There are some 3 or 4 species, maybe, if I remember correctly, from those plantations, which we all of a sudden are finding in the native conifer forest, especially in the sites which are closer to those plantations, like in the coast mountains. Yeah.
And do you know if has anybody looked at the functioning of those introduced AMF fungi? Are they being helpful? Are they being parasitic?
No, we are not there yet. Yeah.
Okay, cool. We have a question from Camille. Thanks, César, for the inspiring talk. Do you think that mycorrhizal collectives can last permanently in the soil despite seasonal variation?
Yeah, that's a good question. I think that to find evidence for that, we would need to for example, in the mice experiment that the Wang people did, in a crop, they will need to came back in the next season or in the next year and resequence the data and also analyze again the phosphatase activity. And if they found the same yields, is an evidence, approximate evidence of the permanence of these collectives in time. Definitely is something that should be done and also analyze more traits that are costly to these microbes, but that are beneficial for the host. So I would say that that's like a next step to improve this concept.
Great. Helen has a question. Did you have 2 publications on the gaps in mycorrhizal research, or was it the original analysis of the EU versus South American publications?
Yeah, two publications. The first one was just like a literature review, but what we didn't analyze. We analyzed in the one with Guillermo Bueno, we just analyzed like the most cited articles and what they were about. But in the second one, we went further and analyzed the functions and the mycorrhizal types study. Yeah, so 2. Yeah.
And I agree with Helen that it was a great idea for an analysis. I was sitting there personally thinking, there are a lot of interesting gaps there. We should do it also for more broadly, I think, within the domain. There is a request for a post for the YouTube channel and the upcoming conference to be posted into this. We can do that later. Another question from Camille. What about hyphal grazing by insects or invertebrates? Have you looked into that?
Sorry, hyphal grazing?
Yeah, hyphae grazing by insects.
No, I haven't looked at that. Interesting. No, sorry, I haven't looked at that. Yeah.
We've been accidentally looking at that in my lab currently. We've had mites everywhere. So I was really curious when you were talking. Oh, Jeff, sorry, there are more questions. I missed those. Jeff Powell, thanks, César. A really enjoyable talk. You are finding a high diversity of AM fungi in your studies. Are you finding many that you would think are endemic? And if so, are the endemic AMF more common in certain environments?
Yeah, that's a great question. There are some few species which are in the vineyard project, which I didn't show the results, but we have analyzed along with Paula. Well, two things. The first is that, as expected, in vineyards with a lot of phosphorus, like 1 ppm or 100 ppm of phosphorus, we have like 3 or 4 species of AMF, and they are always the same. No matter if the vineyard is next to Santiago, so 1,000 kilometers from here, or if the vineyard is here. So, and just 3 or 4 species, which I am not used to that. That's too little. But yes, in the more remote areas, we have a lot of biodiversity in a single plot. There is one study that I have from 2016 where we have 18 species in a Nothofagus plot. So despite being Nothofagus, meaning ectomycorrhizal trees, the understory associates with 18 species of Glomeromycota in one 30 by 30 meters plot. And those are very particular from that site. So yeah, we have some of those patterns as well. Yeah.
Another question, can you please explain the reason why the higher biomass of trees is associated with lower ectomycorrhizae in your preliminary research? And have you encountered the opposite for arbuscular mycorrhiza, i.e. lower biomass for tree?
No, for both type of trees, for ectomycorrhizal trees and for arbuscular trees, we find the same pattern. It's a caution. As I put in the slide, we need to see this with caution because it's just very preliminary result. We think that because the trees that we work with are very old, for example, the araucarias easily can go up to 1,000 years. And Nothofagus, maybe 500, 800, the oldest one. Maybe it's possibly related. I don't know. It's just a suggestion, but maybe it's related because they don't longer need the symbiosis, or they don't longer need it as much as in initial stages. Or it may be related to stabilization of soil conditions. So in that slide, we still need to take into account the variation on soil conditions. So maybe that has a bigger effect on tree biomass than the abundance of ecto and arbuscular mycorrhizal fungi. But yes, for both types, we find the same. And the interesting thing is that in the case of arbuscular, both for Fitzroya cupressoides, this very Fitzroya cupressoides is the only species of the Fitzroya genus, and for the Araucaria, like 400 kilometers north. So it's the same pattern in these two conifers, which I also find interesting.
Yeah, really fascinating. And just following up on that, if I understood correctly, though, when you then went to the world's oldest tree, which was mind-blowingly old, you then found increased richness of fungi in the roots of that tree. So do you have an idea as to why then, when it gets even older, why it then flips?
No, and is really curious. In that slide, I was talking of all fungi, not just AMF, but all fungi in general.
Yep. Okay.
And we need to resequence because with ITS2, of course the results for Glomeromycota were not too exciting, very low diversity. So I need to send the samples to sequence AMF specifically with other markers to see if the pattern holds for AMF. But for all fungi, yes. And it's very interesting because this Alerce milenario, Alerce abuelo, is really lonely. It's like 2 kilometers away in the path from the rest of the forest of the same species. So it's surrounded by other things, by many other things. So yeah, we don't know. So and also that's why we exclude that particular tree in the analysis of diversity of AMF versus biomass, because it's so strange. I think that 20 times. So that one, the weight is 66 tons, and the next a bigger tree is around 5 tons. So it's huge.
Yeah, right. Well, thank you very much, César, for spending time with us today, for talking about your research and the research being done more globally in the South American continent. That's fantastic. Thank you, everyone, for attending, for the questions. Remember, same time, same place at the end of July. We'll see you all later. Thank you.